In this video, we are going to go over the answers for the worksheet of chapter 9 and 10. On part A, we are going to be labeling the parts in a nucleotide. So we need to identify the 3 parts. In part A, this is going to be the phosphate group, as you can see, there's 3. So this is a triphosphate. Part B represents the sugar motif. That in this case, because on carbon 2, we have a hydroxyl, that means that this is a ribose sugar. Part C is going to be the nitrogenous. In other words, the nitrogen base. In looking at the structure, this is guanine. Let's move on to part B. We're going to be filling the blanks using the using the structure in part A. So, the first thing that we need to do is the name of the nucleotide, the structure above. And whenever we have a nucleotide, then we are going to write down the nucleoside name first. And guanine turns into guanosine. But because this is a nucleotide, we need to express how many phosphates we have. So this is guanosine triphosphate. Then the phosphate group is bonded to the oxygen atom on carbon number 5 in the sugar structure or the sugar motif. The functional group that we can see in part A, if I highlight the atoms, is going to be a phosphoric anhydride. Part 4, it says the phosphorus atoms circled in part A of the nucleotide are labeled as certain words respectively. So when it comes to this, I'm going to do it in a different ink. These phosphorus groups are labeled as alpha, beta, and gamma. Number 5 is the name of the nitrogenous base, which I already wrote the answer above. So this is guanine. Understand that the nucleoside or nucleotide name is different from the name of just the nitrogenous base. A blank is the bond that connects the sugar motif to the nitrogenous base. And that is this bond right here. That is called a beta and glycosidic bond. So the beta N is not necessary. Beta represents that the bond is pointing up, N means that it is bonded to a nitrogen. So glycosidic bond or beta N glycosidic bond is also correct. And number 7 says this nucleotide can be incorporated into blank nucleic acid. Specifically, is going to be ribonucleic acid, because as you can see, we have a ribose sugar, so this particular nucleotide can only be found in RNA. Let's move on to part C. So in part C, what we're doing is labeling the following structure based on the words that are given. So when it comes to this, picture number 1, I'm just going to start highlighting them so they're more clear. It is this pentagon, and that is specifically is going to be the oxyribose. And the reason why we know it is deoxyribose and not ribose is because we have double-stranded DNA. Structure 2. This is specifically this. This is a nitrogen base. Number 3 is this whole segment. So this means that that is going to represent a nucleotide. Number 4 represents all of this, which that is a base pair. And because specifically we have 3 hydrogen bonding events, this implies that this is a GC base pair. Number 5, we are comparing the dotted lines to one another. So that's going to be the hydrogen bonds. And lastly, on number 6, we're pointing at this circle right here, which is going to be the phosphate group. Let's move on to part D. On part D, we're going to be writing the correct complementary strand for the following DNA sequences. Understand that whenever we're writing the complementary strand for a DNA sequence, base pairing are going to go in the following manner. Every time you have A, that's going to be base pairing with T, meaning adenine, base pairs with thymine. Every time we have G, that is going to base pair with C. So when it comes to this, understand that, as you can see, I'm just going to write the answers as I go along. So in the first one, we have GGA. Let me do it in a different color so at least I can distinguish. So this is GGA, TAT, AGC, GAG, ATA, TAG, AGA, GTA, TGA, CAC, ACA, GAG, and lastly ATA. Now I'm going to complete the problems for 2 and 3. So in the next one, we have GGC, CTA, AAA, TTA, ATC, GAT, GAT, AGC, ATG, ATG, CAA, CCA, and lastly, CGA. The last one, we have AAT, CTC, GGG, CCA, CTA, GTT, AAA, CGT, GTA, CCC, GGC, CAC, and lastly, TTG. In the last part of the worksheet, we are going to be putting in order the steps in DNA replication. This is not representing all the steps that you guys need to know, but more of a generalized step. So when it comes to DNA replication, understand that the first portion of what is occurring is that one of the first things that need to happen is that we need to separate the parental strands. So here, number one is going to be when helicase binds the parental DNA to start unwinding it, okay? But understand that this is more generalized. The specific steps that you guys need to do are more detailed in the slides. Then we're going to have primase catalyze the synthesis of the RNA primer. Then we're going to have DNA polymerase 3 add complementary nucleotides continuously and discontinuously to the 5' in the 5' to 3' direction in the system. Then we're going to have DNA polymerase 1 replace the RNA primer and is going to place deoxynucleotides. Then the ligase are going to be sealing the DNA fragments in both the leading and the lagging strand. And then lastly, we have the two replication forks meeting in the opposite on the opposite sides of the circular DNA at a specific termination region, and then topoisomerase enzyme is what's going to be unlinking them.